Mechanism of DNA replication. How DNA replicates, or how one DNA molecule that produces two DNA molecules. Requirements for this DNA replication. Enzymes. DNA dependent DNA polymerase, helicases, topoisomerases. Nucleotides. Type of nucleotides: deoxyribonucleoside triphosphates. Deoxyribonucleoside triphosphates. What are those? Example: deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate, deoxythymidine triphosphate. These deoxyribonucleoside triphosphates like deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate, deoxythymidine triphosphate play dual role. One function acts as substratum, another function provide energy. How they provide energy? For example, here ATP, deoxy-ATP. It hydrolyzes into deoxyadenosine monophosphate plus PPI plus energy. This energy utilized in formation of helps in phosphodiester bond. Next, PPI, there is a pyrophosphate. Hydrolyzes into two inorganic phosphates plus energy. This energy helps in formation of hydrogen bonds. So this deoxyribonucleoside triphosphate play dual role. On one hand they act as substratum, on another hand they provide energy for the formation of the bonds in the DNA, new DNA. First they hydrolyze into adenosine monophosphate and pyrophosphate, energy liberated. That energy helps in the formation of the phosphodiester bonds. Again, pyrophosphate, they hydrolyze and produce the two inorganic phosphates and energy. That energy helps in the formation of hydrogen bonds. DNA is the longer macromolecule. During replication, both strands act as template. In DNA replication, Both strands act as template. Both strands are involved in the formation of new DNA molecule. These two strands are not separate at once. In some region only, the two strands separate. Enzyme involved to unwind the two strands in that particular region, the region where the two strands are separate, ORI, that is origin of replication. Origin of replication, the region where the two strands are separated and replication initiated, that is origin of replication. If the OR is absent, OR nothing but that is the sequence of nitrogen bases in the DNA, in that region only the two strands unwinds. For this unwinding, enzyme involved, helicase. The region where the two strands of DNA unwinds and replication initiated, that is origin of replication. Enzyme involved in the unwinding of two strands of DNA, helicases. So in one region, the two strands they are separate. The separated strands looks like fork. So that is called replication fork. Where the two strands unwinds, that looks like a fork. In that region, replication initiated. So that is called replication fork. So replication fork, that is form two sides in that DNA. During replication, the two strands of the longer DNA not separate at once in a single stretch, because it is a completely energy consuming process. That is endergonic process. High energy is required. So because of that, only in some region the two strand unwinds. That region is origin of replication. Enzyme involved in this unwinding of two strand, helicases. When the two strands are separated, tension created in the remaining coiling, that tension reduced by enzyme topoisomerases. Function of helicases unwinds the two strands. Topoisomerases reduce the tension created during the unwinding of the two strands. Both strands act as template. One strand 3' end to 5' end direction. Another strand 5' end to 3 end direction. New DNA strand always synthesized in 5 to 3 end direction only. New strand always synthesized 5' end to 3' end direction only. The template strand is the 3' to 5' end. In that region, new strand synthesized continuously. So it is a continuous synthesis. So the template strand is the 3 to 5 end direction, synthesized completely, continuously, and continuously synthesizing strand called leading strand. Leading strand, the template strand which is the 3 to 5' end direction. New DNA strand that synthesizes 5 to 3 end direction continuously. That is continuous synthesis. And this continuously synthesizing new DNA strand is called leading strand. And complementary strand, another one, 5 to 3' end direction. In this DNA that form, new DNA that is formed, no doubt that the 5 to 3 end direction, but it's a discontinuous. Discontinuous. When it opens, again new strand. Some part or some fragment that is formed. Those fragments which are formed, Okazaki fragments. and that strand is called lagging strand. So leading strand is a continuous strand, lagging strand is the discontinuous strand. Fragments are. That means some part if separate, one strand, one fragment. Again it separate, another strand, another fragment is formed. So discontinuously formed. So that is called lagging strand. Lagging strand is the discontinuous, leading strand is the continuous synthesis. And those fragments which are formed toward the 5' end to 3' end template strand called Okazaki fragments. They are connected by DNA ligase. Okazaki fragments are connected by DNA ligase. It is enzyme consuming process. Enzymes supplied by deoxyribonucleoside triphosphate besides act as substratum, they provide energy also. So finally, the one strand that separate is a 3' end to 5' end direction. New strand that is synthesized, that is in 5 to 3' end direction. Here it is the 3' end to 5' end direction, 5 to 3' end direction. New one that synthesized 5 to 3 end direction. That means new strands are synthesized in bidirectional, one and another one opposite. One strand in one direction, another strand in another direction. So during the replication of the DNA, new strands that are synthesized in opposite direction, but the synthesis is always 5 to 3' end direction only.